This segment is on neuroglia. Neuroglia are the cells in neural tissue that support and protect neurons. Neuroglia are abundant and diverse, and they account for roughly half of the volume of the nervous system. The central nervous system contains a greater variety of neuroglial cell types than the peripheral nervous system. We have only a limited understanding of the functions of these cells. The central nervous system has 4 types of neuroglia: ependymal cells, astrocytes, oligodendrocytes, and microglia. The peripheral nervous system has 2 types of neuroglia: satellite cells and Schwann cells. Ependymal cells line the central canal of the spinal cord and the ventricles of the brain. They assist in producing, circulating, and monitoring cerebral spinal fluid. The central canal of the spinal cord is a fluid-filled passageway that extends along the longitudinal axis of the spinal cord and the brain. Cerebral spinal fluid fills this passageway and also surrounds the brain and spinal cord. Cerebral spinal fluid circulates continuously and provides a protective cushion to the brain and spinal cord, as well as circulates dissolved gases like oxygen, nutrients, wastes, and other materials. The ventricles of the brain are also lined by ependymal cells. The ventricles are enlarged chambers found in several regions of the brain. The ependymal cells that line the central canal of the spinal cord and the ventricles of the brain form an epithelium called ependyma. Astrocytes are the largest and most numerous neuroglia in the central nervous system. These cells have a variety of functions. Many of them are poorly understood. They help to maintain the blood-brain barrier, provide structural support, regulate ion, nutrient, and dissolved gas concentrations, absorb and recycle neurotransmitters, and form scar tissue after injury. Astrocytes maintain the blood-brain barrier. Compounds in the blood do not have free access to the central nervous system. The cells lining the central nervous system capillaries create a blood-brain barrier, and they control the chemical exchange between the blood and the interstitial fluid. These cells essentially isolate the central nervous system from the general blood circulation of the body. The astrocytes have slender extensions that wrap around capillaries. These processes form a complete blanket around the capillaries except areas where the neuroglia come in contact with the capillary walls. Oligodendrocytes have slender cytoplasmic extensions, but the cell bodies of oligodendrocytes are smaller, with few processes than astrocytes. The processes of oligodendrocytes are in contact with the exposed surface of neurons. These processes completely surround the axons in the central nervous system and insulate them from contact with the extracellular fluid. The tip of each process expands to form an enormous pad which resembles a pancake. This flattened pancake gets wrapped around the axon, forming concentric layers. This wrapping is called myelin. The myelin wrapping serves as an electrical insulation and increases the speed of the action potential traveling along the axon. When an axon is wrapped in a myelin sheath, this axon is said to be myelinated. Each oligodendrocyte myelinates segments of several axons. The large areas of the axon that are wrapped in myelin are called internodes. The gaps in between the adjacent internodes are called nodes or nodes of Ranvier. In dissection, myelinated axons are glossy white because of the lipids in the myelin. As a result, regions in the central nervous system that are dominated by myelinated axons are known as white matter. Not all axons of the central nervous system are myelinated. Unmyelinated axons are axons that are not completely covered by myelin. Areas of the central nervous system that contain these unmyelinated axons, as well as the cell bodies of the neurons and their dendrites, both of which are unmyelinated, have a gray color and make up the gray matter of the central nervous system. Microglia are the least numerous and the smallest neuroglia in the central nervous system. These cells are phagocytic. They can migrate through the neural tissue. They form during embryonic development from stem cells that also produce monocytes and macrophages. They migrate into the central nervous system during embryonic development as the nervous system forms. They remain in the central nervous system throughout life, acting as a wandering janitorial service and police force by engulfing cellular debris, waste products, and pathogens. The peripheral nervous system also has neuroglia. There are 2 types of neuroglia in the peripheral nervous system: satellite cells and Schwann cells. Satellite cells surround neuron cell bodies in the ganglia. They regulate the environment around the neurons, much like the astrocytes do. Schwann cells form a sheath around peripheral nervous system. A Schwann cell can myelinate axons, but only a segment of a single axon. In the central nervous system, oligodendrocytes were able to myelinate portions of several adjacent axons. A series of Schwann cells is required to enclose an axon along its entire length. In the peripheral nervous system, Schwann cells play a part in repairing damaged nerves. In a damaged nerve, the axon distal to the injury site degenerates in a process known as Wallerian degeneration. The Schwann cells do not degenerate, and they proliferate to form a solid cellular cord that follow the path of the original axon. As the axon grows alongside the appropriate cord of Schwann cells, it may eventually reestablish its normal synaptic contacts. If it stops growing or wanders off in a different direction, normal function will not return. Limited regeneration can occur in the central nervous system, but the process is much more complicated because there are many more neurons involved, and the astrocytes may produce a scar tissue that prevent axon regrowth across the damaged area. Astrocytes may also release chemicals that block the regrowth of axons.